Good morning, everyone. My name is Gena Wang. I'm a SMID-cap biotech analyst at Barclays. That would be a big mistake. It's my great pleasure to introduce our next speaker, Matthias Jaffe, Chief Financial Officer from Logic.
Thank you, Gena. Thank you for the opportunity. Thank you, Barclays, for an opportunity to tell you the story of Logic Bio in this very intimate setting here. Logic Bio is a genetics medicine company with a focus on pediatric patients. And I think the key is the pediatric focus. There's a lot of excitement these days about genetic medicine. We have approved drugs. We have a number of clinical trials, a lot of really interesting results that promise therapies and cures for a lot of patients. But a lot of this activity is restricted to adult patients. And pediatric patients are being left out. Even though of the 6,000 or so monogenic diseases that could potentially be addressed with genetic medicine, 70% of those affect children, and many of those affect children with a very early onset. And these children don't always have the luxury of waiting until adulthood to be treated, simply because in the worst case, they won't make it to adulthood. And in the cases where they do, they have accumulated such severe developmental issues that cannot be reserved, that even if they were treated as adults, it really wouldn't it wouldn't have the same impact as if they were treated earlier. So this is where Logic Bio comes in. We believe that we overcome a number of those shortcomings of other approaches, which usually fall into two categories. It's either a lack of durability, which you see with genetic therapies, with gene therapies, which we often refer to as gene transfer, where you take a healthy gene under the control of an episome, you bring it under the control of a promoter, you bring it into the cell as an episome, and it will have a positive effect, but it will be diluted out as the cell replicates. It will either be lost or it will simply be diluted over time. And then you have the issue of safety. When you talk about, again, gene therapy, an exogenous gene under the control of a promoter, there's always a risk of non-specific integration with AAVs, and if these AAVs integrate in front of an oncogene with an exogenous promoter, you have the risk of inducing cell proliferation and therefore cancer. That is something that luckily we have not yet seen in the clinic, but we have seen extensively in preclinical models, and there's really no reason to believe that that risk does not exist also in humans in the clinical setting. And especially if you're going into children, it is something that you want to avoid by all means. On the other side, you have gene editing approaches, which rely on nucleases. And this is another source of safety concerns, especially if you were to use them in vivo, because as specific as they are not as specific as you would want them to. And there's always a risk with nucleases to not only have immunogenic reactions, but also to have non-specific modifications that you cannot control. So for these two reasons, we believe that children are underserved, and this is where Logic Bio, with its GeneRight technology, comes in, and we believe offers a unique alternative. And that's what I want to talk to you about today. GeneRight, our technology, and LB001, our lead indication for the treatment of methylmalonic acidemia. So I will be making forward-looking statements. I advise you to review the risk factors that we have disclosed in our S1 and our 10Qs. But let's dive in with GeneRight. So again, I told you all the drawbacks that prevent other approaches to go into pediatric patients. GeneRight is a proprietary approach that was developed at the labs of Mark Kay in Stanford, and to which we have all the required IP. It is an approach that is nuclease-free. We do not require exogenous nucleases. We do not depend on exogenous promoters. We are very site-specific, and I will explain how all of this works. And finally, we do integrate into the chromosome, therefore providing a durable benefit to the patient even in rapidly dividing cells and growing tissues. Before I explain the details on the molecular level of how GeneRight works, let me tell you a little bit about our pipeline. As I mentioned, LB001 is our lead program for the indication of methylmalonic acidemia. That's a very severe disease, no treatments, very early onset. I'll talk a little bit more about that. We're in IND-enabling studies at this time. We expect to file an IND by late this year, and then we intend to start our phase 1/2 clinical trial in pediatric patients soon thereafter, and hope to get clinical data in 2020. We actually have proof of concept in 3 other indications, hemophilia B, alpha-1 antitrypsin, and Crigler-Najjar. This is, I think, important for a number of reasons. It talks about the modularity and the platform nature of our approach. It also talks about the robustness, because all of these 4 proof of concepts have been generated by 4 different academic groups and have been independently published in peer-reviewed papers. So the technology has been proven in mice models, and we are now going to prove it in humans. We are unlikely to take one of these other 3 indications as our next second indication to be developed in-house. There's a lot of interest from Big Pharma to potentially partner it, but we believe that our second indication is going to come out of our discovery engine process, where we're looking currently at a number of indications, all of which have a number of characteristics in common. They have a high unmet need. They have a very early onset. There's a potential for making a big, dramatic difference in the lives of the patients. They're very well understood from the biology perspective, and they're animal models that allow us to test our hypotheses early on. There's really no reason why that second indication needs to be a liver-targeted approach as LB001 for MMA. However, we believe that in order to risk mitigate the process of expanding our pipeline, we will go into the liver with probably the second and third indication, simply to be able to maintain as many pieces of our technology common as we can. So as long as we stay in the liver, we will continue to rely on our capsid, LK03, which we believe is best in class as a liver-tropic capsid. We're going to continue to integrate into the albumin locus with similar homology arms, and therefore derive similar integration rates and similar expression rates. However, we believe that there's no reason why we cannot go into other tissues, and we have data and we have experiments that we have undertaken where we have integrated using the same GeneRight approach into other tissues, into locuses that are under the control of other promoters, and basically you simply have to play with the components of the platform, change the capsids, change the homology arms, but there's really no reason why this wouldn't work. This is probably something that we're going to be working on for our 4th or 5th indication. But let's dive into GeneRight at this point. So at the molecular level, our constructs, our GeneRight constructs, are single-stranded DNA that are capped on both ends by standard ATRs. And then you have, in blue, homology guides. These are sequences that are between 500 and 1,000 bases long, and that are homologous to the albumin gene. And they are designed in such a way that they integrate just upstream of the stop codon. In orange, you have the transgene, which in the case of LB001 is the MUT gene. That's the gene that is mutated or defective in 70% of all MMA cases. And these homology arms will then align or anneal with the native albumin locus. And through the process of homologous recombination, the MUT gene will be incorporated into the chromosome just upstream of the stop codon. Again, the important factor here is to understand that this is happening without the use of exogenous nucleases. This is a natural process based on homologous recombination. It is admittedly a modest efficiency process. The integration rate is probably in the rate of 0.1 to 1%, median of around 0.5%, but it's highly specific and a very high fidelity process. Once you have integrated the transgene into the chromosome and have stably incorporated into the genome, you will produce a polycistronic messenger RNA. That messenger RNA will contain the full sequence of the albumin gene, together with the full sequence of the MUT gene just ahead of the stop codon. And at this point in time, I'd like to explain what that little pink segment is with the 2A. That's 2A peptide. That's a technology that is derived from CAR T applications. It comes from the foot and mouth virus. It has gone through the FDA. It has been proven to be safe. And what it does, it's a self-folding sequence that as the ribosome comes and reads through the messenger RNA, it will effectively stall as it encounters the 2A sequence, stop, and then continue right afterwards. So basically, as the ribosome goes through this polycistronic messenger RNA, it will produce equimolar quantities of both genes. You will have the albumin gene, the albumin protein, and the MUT gene derived from a single messenger RNA. There are a couple of important things to mention here. First, you can obviously have any signaling sequence that you want in your transgene. In the case of MUT, it's an intracellular protein. But in the case of factor IX, it's a secreted protein. So we have full autonomy as to which compartment we want to send the proteins into afterwards. And this second step, after I indicated that we do not rely on exogenous nucleases, I want to point out, shows how we can do this without the use of exogenous promoters. So again, the construct itself, if it was kept as an episome in the cell, would not be transcribed, would not generate any protein. It needs to be integrated so that it falls under the control of the albumin promoter and get transcribed. And the genius of our founders was to marry this very high fidelity natural process of homologous recombination with a very strong promoter of albumin. So effectively, you're buying all the good features of homologous recombination, and then you're paying for the one downside of homologous recombination, which is the modest integration rate compared to, let's say, when you actually use a nuclease and you do a double cut of the DNA and need to repair it, and then making up for that with a very strong promoter. To underline this a little bit more, I'm showing here the 2,000 most highly expressed genes in the liver. And right against the y-axis, number one is albumin, by actually order of magnitude higher than most other genes. Actually, if you see a line that is below it at 0.1% of the albumin expression level, you still see that about 95% of all genes are expressed at below that level. So it's an incredibly potent gene. And again, to view that from another perspective, in the middle graph, what you see here is albumin expression as transcript per million compared to some genes of interest. For example, PAH for PKU, factor IX for hemophilia, MUT, which is our transgene of choice for MMA, and then for alpha-1 antitrypsin. And you just see it's orders of magnitude lower. Actually, at the very bottom of the albumin bar, you see a little orange bar, which corresponds to 1% of the albumin expression. If you blow that up on the right-hand side, you will see that 1% of albumin is about the same physiological level as a PAH and factor IX, and between 5 and 20 times more than MUT and alpha-1 antitrypsin. So again, if there's one thing I would like you to take with you today is that you don't treat a patient with integrated DNA. You treat a patient with expressed protein. And the one thing that's important here to understand is that the integration rate times how much stronger the albumin promoter is than the natural promoter of your gene is what gives you the final number, which is the expression level of the protein, which is what really matters. And in each of the 4 proof of concepts that we have published on, we have generated enough protein to have therapeutic effects that have rescued the phenotype of the disease models. So once again, very quickly, our gene-right construct has the homology arms in blue, the transgene in orange, and the capsid. Simply by changing the transgene and leaving everything else, we have 4 proof of concepts. We believe that most, if not all, liver-targeted approaches can be used we can take the same approach by simply taking the transgene and swapping it out. This is really a feature of our platform character, where we believe that we can very quickly go from first indication to second indication because we just have a lot of things that are common. This will help us from the safety perspective. This will help us from the FDA review perspective. This will help us from the CMC perspective. And it highly de-risks the approach. You already know where you're going in. You know with which rate you're going in. You know with which rate you're going to be expressed. And the only thing you're changing is what is it you're expressing. And you have chosen that transgene based on the biology of the disease that you want to treat. So I think this is going to allow us to quickly move from one indication to the other. As I mentioned, if you change the homology arms to target at different loci, and you change the capsid to target a different tissue, you can do the same thing for other indications in other tissues. We have actually 2 papers in keratinocytes where we have proven that you just simply choose a different locus and you can do the same thing. We believe that the same approach can be used for muscle and for CNS and basically any other tissue of choice. Capsids is another feature that is really important for Logic Bio, and our IP is not focused exclusively on the GeneRight technology. We also have a lot of our IP around a portfolio of AAV capsids that also come out of the lab of Mark Hay in Stanford. Specifically, we're currently working with LKO3, which is a capsid that was derived out of a combinatorial shuffle of AAV vectors that were then screened in a mouse that actually had a humanized liver. Basically, the liver of the hepatocytes were ablated and where the liver was repopulated with human hepatocytes. So you're using a human liver to scan and to screen for the viruses with the highest transduction rates. And that's how we found LKO3. And we believe that currently LKO3 is the best-in-class liver-directed target for humans, as opposed to many of the other AAVs that have done really well in mice, but don't really translate that well into human tissue. So much for the technology. Let me talk a little bit about MMA. So MMA is a devastating disease. It basically results from the mutation of the MUT gene, which does not allow the patients to properly metabolize branched-chain amino acid or short-chain fatty acid. Even though there's neonatal screening for it, most mothers know that there's something terribly wrong with their children even before they get the results back. There is no treatment. The only standard of care is to severely reduce the protein content in the diet of these children. And when I mean severely reduced protein content, it's basically an all-carbs diet. You're playing a very difficult game of trying to get these children to grow while at the same time not giving them too much to eat that would lead to a metabolic decompensation. Very few of these patients survive their second decade. The median age of survival is 2 to 3 years. Those that do survive almost all have to go through a combined liver-kidney transplant. It's effectively a devastating disease. What we have done is we have used a number of mouse models to test our approach. There are different severity levels of these mouse models. We have used a very severe model where the animals that are untreated die within hours of being born. As my scientific colleagues like to say, it's really hard to do experiments when your control group is all dead. So we have moved to less severe models. This is data that was actually produced at the NIH by Chuck Venditti when one of these less severe models where the animals do survive. But as you can see in the left panel, the treated animals that have received a single injection of LB001 or the murine equivalent, after 1 month have put on significant additional weight. And what is important to understand is that even though they have put on more weight, obviously as a consequence of consuming more food, the metabolites that usually accumulate in diseased animals are significantly reduced. So if a diseased animal would eat more, they would have more of these metabolites or intermediates. Here we see a significant decrease. In the more severe models, we have actually shown survival up to a year of animals that otherwise would have died within a couple of days. What's really surprising, though, is that Chuck, who has worked with a number of other groups on different approaches to treat MMA, had seen in the past that when you do regular gene transfer, where you take the MUT gene, you introduce it into the cell under the control of an exogenous promoter, that these animals, after a year, up to 70% of them will develop hepatocellular carcinoma. Again, going back to what I said earlier, that there's always some nonspecific integration of AAVs that can lead to cancer. So he was interested to see what happens here. So after waiting for 1 year, he opened the animals, and in the middle panel you see a slice of a liver, and the darkened points are cells that have been transformed. Those are cells that have incorporated the MUT gene as a consequence of GeneRight. On the left panel, you will see a slice of a liver after 1 year, and you'll see you don't have to be a pathologist to realize that there's significantly more transformed cells than before. Now, the question is, how can that be? You have not given these animals any more GeneRight construct, yet you have an increase from about 1% to over 25% of cells transformed. And the reason behind this is that the cells that have been transformed have a competitive advantage. Not only are they able to properly metabolize the amino acids and the short-chain fatty acids, but the product of MUT at the end of that step is succinyl-CoA. And succinyl-CoA goes into the Krebs cycle. The Krebs cycle produces ATP. So the cells that do not have MUT cannot have a proper energy metabolism. The cells that do are actually much more healthy. So what you see is that as the liver grows over time, the cells that actually have received the transgene have a competitive advantage over those that did not and outcompete. So what you see here is the opposite of what you see in gene transfer experiments, where over time you're diluted out. Here, over time, you have an increase in the total amount of cells that are expressing your protein of interest. Another thing that I would like to mention here is often people say, well, how do you know that with such a strong promoter you don't end up with too much protein? This will not apply for every single model, but at least in this case, it's clear that if it wasn't beneficial for the cells, you wouldn't have the competitive advantage and you wouldn't have the expansion. So it's not only safe for the cell, but it's actually advantageous. And this competitive advantage is a feature that we would like to build into our second indication, and there are a number of models and diseases where this would apply. So to wrap it up, we believe that we're a first-in-class genetic medicine 2.0, you could say, approach, where we have a number of benefits, both in the dimension of durability as well as safety. We believe that our platform is a true product engine that will allow us to very rapidly go from one indication to the other by leveraging all of the experience and fundamentals that we have around the technology. I think we have a really good team. The team is growing. We're expanding. We just moved into a new location. We have proof of concept in a number of different animals. Our MMA program is expected to file for an IND at the end of the year. We expect to have data early next year. We just IPO'd a few months ago, and we believe that this next year is going to be a key year for us, allowing us to take GeneRight into the clinic for the first time, and in 2020 prove that what has been true in a number of different mouse models also applies to humans in the same way.
Thank you, Matthias.
Thank you.
I think we are running out of time. I would just ask one question.
Sure.
Regarding, I think you comment, you can easily switch to different indication by, swap the vector and promoter and the transgene, right? So just wondering, what will be the criteria you will be looking at, initial editing efficiency, and then how that apply to the specific indication you are looking for?
The criteria for the editing efficiency?
Yeah.
So I think initially we will stay in the liver. I think we will learn a lot from the liver, and that will allow us, knowing what the efficiency has been in MMA, to make extrapolations as to what other diseases can we treat with that same level of expression. I think as we move into other tissues, we will have to use those findings and marry integration level efficiency with expression levels driven by the specific promoter and the transduction level of our new capsid into those types of cells to decide, what indications will allow us to express enough protein to treat the phenotype.
Thank you. Maybe just one quick question regarding the MMA. You will have an IND filing later this year, right?
Mm-hmm.
So any thoughts on the first phase 1 clinical trial design?
Well, the protocol has not been finalized. It will be submitted with the IND. Just a month ago, we brought on board Ken Hutton as our VP of Clinical Operations and Development. So I think there's a number of questions that will have to be answered, but what we know for sure is that it's probably going to be a small trial, probably somewhere in the order of 8 patients. We are targeting pediatric patients, so in the 0 to 12 years of age, all of them MUD0. And we'll be monitoring a number of different parameters, weight gain, levels of metabolites, and I didn't mention this, but obviously in the cells that have been transformed, the albumin will be modified by having a little residue of the 2A peptide attached, so we can easily measure the ratio of 2A modified albumin to natural albumin and get a sense for the levels of integration. We tend to see 2A albumin in mice after 7 days post-treatment, and we believe that we should be able to very quickly, therefore, in patients also, get a number of parameters that will allow us to know whether we have been successful or not. Regarding timing of data, I think it will depend a little bit on how quickly we can recruit patients and how much data we want to have in hand before we start disclosing. I think it's unlikely that we're going to disclose the first data point of the first patient. So once we have enough data to make a convincing case, we will disclose that data.
Thank you very much, Matthias.
Thank you.
So we will have a breakout session at the Ponciana 2.